Oh, we're good? Wait, Rick, that's fine. I'm going to sit, okay. Do I ring the bell?
Ring the bell.
Oh, okay. Is there enough people here? Yeah, that's fine. Ready? So, I'm so sorry. I didn't know it was fine. Oh, man.
Okay, welcome everyone. I'm Rick Starr, and we're here today to listen to Molly Alvino talk about her thesis work. And the way this works for those of you who are new, we'll listen to Molly for a while. Some might say a long while, and then our committee, Scott Hamilton, Geedy McDonald, and myself, will take her into a little dark, hot, stuffy room and ask her all the hard questions that you don't ask her at the end of her time here. And then we'll come back out and enjoy the celebration. And before we start, I want to acknowledge that her mother, Carrie, and sister Shannon are here in the room, and her dad and Chris are online, we hope, on the and brother watching to cheer her on. So I want to introduce Molly by starting out by saying that she's been here 3 years, which is a rapid time for those of you who are in Moss Landing, and she's been on the go the whole time here. And when she's not here, she's flying to the Maldives or somewhere like that to go scuba diving and have fun, and I'm always jealous of that. So I've had a hard time keeping up with her, and you can understand, whoops, sorry, I've got because she was destined to be a marine scientist. This is a picture of her as age 3 collecting shells in Martha's Vineyard. So she spent a lot of time outside and in the water. As a teenager, she worked in the New England Aquarium in Boston, and much like the aquariums here, Nearshore fish tank, she fed fish and cleaned the tanks, and she completed and logged 70 dives while there. So that's a pretty impressive resume for a young girl. However, I've also heard that she might have gotten bitten by an inquisitive parrotfish. So ask her to show you her scar sometime. Uh-oh. Hang on.
Oh, they're good.
There. So we're back. Okay. Afterwards, after that time in the New England Aquarium, she spent a few months in August to December of 2017 at the Bimini Biological Field Station as a shark lab intern in the Bahamas. And she learned a lot of research techniques there. A lot of folks have gone through that lab, and they do a lot of good work there. She learned how to catch fish on long lines. She's learned about acoustic telemetry techniques and baited remote underwater videos, which was good because that's a lot of what we've done here. When Jake gives us his thesis presentation, you'll learn a lot more about that. That'll come up really soon. And then she also learned how to work up sharks. Here's a picture of Molly with some baby nurse sharks, and I'm just wondering if she has any scars from those nurse sharks. And another thing that I've noticed about her is she early on has been adopted by major sponsors like Nike. And while there, we have this picture of her chasing after a hammerhead, and I'm wondering if she's got more scars from that. Maybe they're just mental. But clearly she's got no fear in the water and is inquisitive. And then you can help me out here. Eleuthera
Yeah.
is an island school in the Bahamas, and she earned her dive master certification there and taught divers from the local high schools how to dive. So here she is wrangling a turtle. I presume you're tagging it or just playing with it?
Yep.
One or the other. And is quite comfortable, clearly, in the water. So I find it interesting that she graduated from Northeastern University in Boston, and on their website it says, "Learn in an urban oasis in the heart of America's college town." So she's in downtown Boston, but I get all these pictures from her from faraway parts of the oceans, mostly in the tropics. So I'm wondering if you really went to school there or not.
I don't know.
You know, but you got good grades there. It's just a matter of whether they're real or not. Because as far as I know, this is what she spends her time doing. Here she is in Baja with a yellowtail, and I don't know the timing of this, but I suspect it was when you should have been in college. And then she talked to me, contacted me in 2021, and wrote in her prospectus that she became intrigued in learning about fishery management and gathering information to prevent fishery collapses, and she thought that Moss Landing's history and reputation for a collaborative environment and close-knit community was really interesting to her. And she came out, looked at the lab, and decided to come here to our delight. So she's been here 3 years and spent a lot of time at sea in those 3 years. It's been very active in the CCFRP program, where she's captured fish, she's tagged fish, she's measured fish, she's worked as a volunteer angler coordinator, she's spent a lot of time entering data, and has learned thoroughly the CCFRP program and protocols. She's been a research assistant in our lab and worked on Scott's ocean acidification project and the search zone monitoring, and has worked with our BOSS system looking at deep sea videos of fishes, and worked on the mini lander project that Jake's doing his thesis on. And she's been very active getting grants and awards. So I've been trying to get her to give me some of that money, but hasn't come my way yet. But she's been really successful in that. So, as I said, that she's been really, really busy for 3 years, and this is what it gets you after 3 years. She's earned time to sleep on the deck. And she's going to talk to us today about her thesis related to the barotrauma that she looked at all those years working with CCFRP. So Molly, you're up.
Okay. thank you so much for that introduction, Rick. And thank you, everyone, for being here either in person or online virtually. This is my defense presentation for my thesis titled Swim Bladder Morphology Influences the Responses of Nearshore Rockfishes to Barotrauma. So first I'll begin with an outline of my presentation. I'll start with the background, then move to the objectives, then the methods and results, and then ending with the discussion. So throughout my presentation, there will be an outline banner across the top of every slide with a vermilion following along. So if you ever get lost, just look to the red rockfish. So starting with my background, so why did I choose to study rockfish in the first place? Rockfish are widely distributed and abundantly found throughout the North Pacific. There are over 60 species in California alone, and these species can occur at a range of depths and habitats. For example, some species can occupy nearshore rocky reefs or kelp forests, while others can be found in deep water slope and shelf habitats. This genus is known to be slow-growing and late to mature. Additionally, due to their abundance, they are prevalent in both the California commercial and recreational fisheries, with more than a couple million fish caught in the fisheries every year. Focusing on the fishery, rockfish do occur in mixed species assemblages. This is disadvantageous because a lot of the gear typically used to catch rockfish, such as trawl nets and hook and line fishing, is non-selective. That means even if you want to catch a vermilion rockfish, you might drop your line down and come up with a gopher instead. So you really can't control which fish or which species you catch. Because of this, there are high discard rates of species that are unwanted or that are deemed too small to keep. Due to rockfish morphology, these fish that are discarded often die as they succumb to thermal stress, and they are, so due to this, they often die, which results in high discard mortality rates, or DMRs, occurring in both the recreational and commercial fishery. Highlighting physiology, rockfish have an internal organ called a swim bladder that is used to adjust their buoyancy. The gases within the swim bladder rapidly expand when fish are reeled up to the surface, and these expanded gases remain in the swim bladder even after time of capture. So the trapped gases often act as a life jacket, and if this fish is thrown overboard, it's so positively buoyant that it's not able to swim down to depth on its own. So these fish, like I mentioned, often succumb to thermal stress, and there is an increased susceptibility to predation by marine mammals and birds, resulting in high discard mortality rates in rockfish. So about half of all bony fishes have a swim bladder, and the swim bladder allows for a change, or allows fish to change and maintain their position within the water column without exerting extra energy by swimming. And there are two types of swim bladders. There's physostomus and physoclistus. And a physostomous swim bladder, as seen in the top figure, has a pneumatic duct that connects it to the esophagus. Fish with this type of swim bladder, such as herring, trout, and catfish, are able to swim up to the surface and gulp air to increase their buoyancy and burp air out to remove excess gas. On the contrary, rockfish have a two-chambered physoclistus swim bladder. This type of swim bladder lost its connection to the esophagus during development, and because of this, it is closed off from the external environment. The loss of the pneumatic duct seen in physoclists results in slower rates of gas resorption and gas secretion, resulting in greater susceptibility to pressure-related injuries. So the loss of the pneumatic duct and the connection to the external environment adversely affects the rate at which gas can enter and exit the swim bladder. Instead of gulping and burping, gas is secreted into and resorbed out of the swim bladder through a fairly complex diffusion process. Gas resorption is controlled by a structure inside the anterior chamber called the rete mirabile, as you can see in the top figure, and then the oval in the posterior chamber controls gas resorption. So this lower diagram demonstrates the process of gas secretion. As you can see, it is fairly complex. There are different components of the building up of gas partial pressures, blood acidification, and a countercurrent exchange system. So the rete mirabile is developed to secrete gases at very high pressure, allowing shelf and slope species to perform gas exchanges even at really extreme depths. But despite all these adaptations, this gas exchange process is incredibly slow. So due to this slow gas exchange, gases that are present in the swim bladder during capture expand with a rapid decrease in pressure from seafloor or from the capture depth to the surface. And this can be attributed to Boyle's law. So in a closed system, pressure 1 times volume 1 is equal to pressure 2 times volume 2. This means as pressure decreases, volume will increase at a rate that is proportional. So this process can be demonstrated by this rockfish. So any injuries caused by a rapid change in pressure are called barotrauma. So the symptoms of barotrauma can present themselves both externally and internally. And many of the external injuries are displacement injuries. So when the swim bladder expands within the body cavity, it displaces other internal organs outward. This and the expansion of gases within these internal organs can lead to injuries such as the stomach protruding in or out of the mouth, anal protrusion, and bulging eyes. So the expanding gases also leak into other areas of the body and usually follow the path of least resistance, which is moving up towards the head. So this, in the presence of densely packed oxygenated blood vessels in the eyes, also can lead to eye crystallization. So barotrauma can also present themselves as internal injuries. And these injuries can't be assessed until the fish is retained and dissected, but these injuries can include a torn or ruptured swim bladder, arterial embolism, and subcutaneous gas bubbles. So all of these internal and external barotraumatic injuries can lead to mortalities, and I wanted to learn more about the causes of mortality and what influences the change in susceptibility to barotrauma in different rockfishes. And so while all rockfish do have this physoclistus swim bladder, barotrauma affects all species differently. So after being from the East Coast and in moving out here, I'd never seen a rockfish before ever. So when I was first brought out on my first CCFRP trip, I noticed that we were throwing some fish overboard and that were able to swim down to depth, no problem, where we had to manually descend some of these fish in order to increase survivorship. So I observed that the fish that we were throwing overboard that were able to swim down to depth were more semipelagic species, but the species that we had to manually descend were more benthic or more associated with the seafloor. And despite being in the same genus, I was interested in seeing if differences in swim bladder morphology or life history traits were driving this varied susceptibility to barotrauma. And just in general, rockfish are pretty ideal study species. So they have a more recent radiation than other teleosts or bony fishes. And the first rockfish was thought to have originated around 8 million years ago, but more than 30 species have evolved within the last million years alone. Because of this frequent radiation, species have been able to develop distinct characteristics and life history adaptations, thus driving the speciation within the genus. So most of the fishes that I am studying, or the 4 species that I looked at, are recently radiated and are more genetically diverse, which may also influence the difference in barotrauma response. So now moving on to my goals and objectives. I had 2 main goals for this study. So my first goal was to determine if there is an association between a species' habitat zone, or where they live, in body size, and the morphological features of their swim bladder. My second goal was to understand how swim bladder morphology and other factors affected susceptibility to barotrauma. So to accomplish these goals, I had 4 objectives. My first objective was to determine the differences in swim bladder morphology among mature, semipelagic, and benthic rockfishes. So I decided on only mature individuals in order to control for the effects of ontogeny and to ensure that the morphological changes associated with maturation would not affect the comparisons made among the rockfishes. So I hypothesized that benthic rockfishes would have thicker, more rigid, and durable swim bladders, whereas semipelagic species would have elastic, flexible swim bladders that are better suited for gas resorption. And my second objective addressed the effect of body size on swim bladder morphology. and I wanted to determine the differences in swim bladder morphology between semipelagic and benthic rockfishes of different lengths. By using rockfishes at a range of lengths, I was able to directly assess the effect of ontogeny and how different stages of maturity affected the development of the swim bladder. I hypothesized that smaller rockfishes will have thinner, less developed swim bladders that are more susceptible to tears and embolisms. And on the other end, I hypothesized that larger rockfishes will have swim bladders that are more robust and have vascular features that allow for more efficient gas resorption rates. My third objective took all of the swim bladder measurements that I used for Objective 1 and Objective 2 to evaluate a relationship between swim bladder morphology and external barotraumatic injuries observed upon capture. So for this objective, I hypothesized that rockfish swim bladders with thinner, more elastic membranes and more developed posterior chamber networks will suffer fewer and less severe barotraumatic injuries. Here are images of two swim bladders from two different rockfish species. On the left-hand side, you have a swim bladder from a blue rockfish, and on the right-hand side, you have a swim bladder from a vermilion rockfish. And I predict that the morphology needed to lessen barotrauma susceptibility will be found in species with similar morphology to that of a blue rockfish. So the fourth objective looks to determine which factors outside of swim bladder morphology may have the greatest influence on susceptibility to barotrauma. This objective was included to kind of look at the more overall picture of barotrauma in rockfishes, as swim bladder morphology may not be the only factor affecting how rockfish deal with these rapid changes in pressure during capture. So I predicted that one or more of these 5 factors will have a significant effect on the incidence of barotrauma, thus contributing to differences among individuals. So then moving on to my methods, I looked at 4 different rockfishes for the study, 2 semipelagic species and 2 benthic species. My 2 semipelagic species were blue rockfish and olive rockfish, and my 2 benthic species were gopher rockfish and vermilion rockfish. So in addition to including their scientific names up there, I also included their length at maturity, or the L100, and the maximum age. And this is just to show that even within a group of rockfish this small, there are already differences in life history traits, and there's just a lot of variability in this genus. So in total, 112 rockfish were collected for this study. All fish were caught via hook and line by volunteer anglers aboard commercial passenger fishing vessels, or CPFVs. And the collection locations included sites around Pigeon Point in the north and Point Lobos in the south. And because I didn't have a biogeographic aspect to this process, these sites were chosen basically out of convenience. And I just ensured that all fish were captured in waters less than 50 meters in order to limit lethal barotraumatic injuries. And fish were collected on a total of 11 fishing trips ranging between January and October of 2023. So after a fish was reeled in by an angler and it met the species and length requirements of my study, the fish was identified to species and a total length measurement was taken. Additionally, all external barotraumatic injuries were observed and recorded, and a T-bar tag was inserted in the muscle at the base of the dorsal fin for ease of identification. And a GPS waypoint was also taken in order to get a rough estimate of bottom depth at time of capture. So in total, there are 4 categories of barotraumatic injuries that I looked at. Within each category, there are either 2 or 3 levels of severity. So my whole goal was to quantify these physical observations that I was observing and quantify them in some way that could be used for later analyses. So to quantify these physical injuries, each injury corresponded with a barotrauma score based on the severity. And a score of 0 indicated that no injury was present, whereas a score of 1 indicated the most severe injury was present. And all rockfish received a score for every category of injury, and the scores were then total for a total barotraumatic index, and this could range from 0 to 4. So back on the boat, after all fish were measured and tagged, they were placed in 5 or 9 gallon stainless steel portable hyperbaric chambers filled with fresh seawater. So after all fish were loaded into a chamber, it was pressurized with pure oxygen to about 37 psi, which is equivalent to about 15 meters depth. This rapid recompression in the chamber simulated a fish being manually descended to at least half of its capture depth. Following the recompression, the pressure was then reduced by 30% every hour to create a stepwise decompression process that allowed fish or gave fish time to adjust their buoyancy after every pressure change. So in total, fish were in these chambers for about 8 hours, give or take a couple of minutes, with 2 gas exchanges during this time. So I would bubble off, use carbon dioxide at a rate equal to the addition of pure O2. So each chamber held between 3 and 6 fish, depending on the size of the fish and the size of the chamber. And this portion of the process was made possible by Joe Welsh, who generously let me borrow these hyperbaric chambers for over a year. So that was really, really great of him. So why did I convince everyone to let me bring these very, very heavy hyperbaric chambers onto 11 different fishing trips? So these hyperbaric chambers were used to reverse any barotraumatic injuries that were experienced during capture. So by quickly recompressing these fish, it would, the expanded gases within the swim bladder would condense, thus reversing any external displacement injury. Additionally, I wanted all fish to undergo the same change in pressure post-capture, but most importantly, I wanted to ensure that all swim bladders were neutrally buoyant at the time of dissection so that my morphological measurements were not influenced by overexpanded or overstretched swim bladders. So after the 8-hour decompression process, all fish were unloaded at the Shore Lab at Moss Landing Marine Labs, where they were placed in deep flow-through tanks. So as you can see in this photo, I placed a few cinder blocks or tunnels at the base of each tank, so any fish that was still a little positively buoyant or a little floaty had a structure to wedge itself under so they wouldn't float up to the surface. Fish remained in these tanks for anywhere from 36 to 48 hours or until neutral buoyancy was reached, and they could swim upright off the bottom of the tank. So after they reached neutral buoyancy, they were euthanized with a lethal dose of MS-222 and placed on ice before being transported back up here to the main lab for dissections. So all fish dissections took place in the necropsy room. Before dissections began, I set up all my necessary tools, which you can see in this right photo. And to begin dissections, I measured the total body length and fork length of each fish and took a full body length a body weight measurement. So after all that was done, and I opened up the fish and kind of removed all the internal organs, I would take tearing threshold, or the amount of air or pressure needed to tear the swim bladder membrane. And this measurement was taken while the swim bladder was still inside and fully connected to the internal body cavity. So when coming up with my methods and kind of figuring out what I wanted to measure, I came up with this. And there's not a whole lot of literature on how to burst or explode a swim bladder. So I kind of had some creativity in coming up with my methods for this. So I knew I needed some sort of pressure gauge and some sort of manual hand pump. And that brought my mind to a blood pressure cuff. So I kind of jerry-rigged this blood pressure cuff. I added a T-bar connector and some additional tubing with a hypodermic needle at the end. And that is the contraption you see on the right-hand photo. So after that was all situated, I super glued a little piece of rubber, as you can see in the left-hand photo, to the membrane to kind of act as a little bit of reinforcement. And it would also make sure air didn't leak around the needle puncture point. So then I slowly added air to the swim bladder and recorded the pressure at which it ruptured or tore. So again, as you can see in the right-hand photo, I took a slow motion video of this whole process and specifically of the pressure gauge, so I was able to get an exact measurement of when this swim bladder ruptured. And this measurement was only taken on swim bladders that were fully intact, so any swim bladder that ruptured or tore could not be included in this because it could no longer hold any air. So that was that. So after tearing threshold, I used great care and a lot of scalpel use to remove the swim bladder from the body cavity. And I then took tensile stress or elasticity, which is the amount of planar or two-dimensional force needed to pull a cross-section of the swim bladder membrane, or needed to pull apart the cross-section of the swim bladder membrane. So like I mentioned earlier, there are two chambers in a physoclistus swim bladder. And the way the swim bladder is, so the posterior chamber fully encapsulates the anterior chamber. So by taking a cross-section at the location I did, it ensured that both the anterior and posterior membrane would be included in this cross-section. So from the cross-section, I took a 3 by 0.5 centimeter sample. And then using hemostats, I kind of situated both ends and pulled the sample along the length of the ruler until it tore, and then the total change in length was measured, and that was my elasticity or tensile strength measurement. So next, I measured membrane thickness. So from that same cross-section, I took an additional sample and placed it in a histology cassette. This cassette was brought up to Diagnostic Pathology Medical Group in Sacramento, California, where the tissue was then cut into 4 equal sections, embedded in paraffin wax, sectioned at 10 micrometers, and stained with hematoxin and eosin before being sent back to me on a microscope slide. So I viewed and photographed these slides under a light microscope and used Image J for processing. So I took 5 width measurements from 3 of the 4 cross-section samples and averaged the widths together to get a final membrane thickness value. So I did this for both anterior and posterior membranes. The anterior membrane appeared as a more solid pink color, whereas the posterior membrane appeared almost striated, thinner, and a deeper purple color. So for most samples, I was able to differentiate the 2 membranes, but for samples that I could not, it was excluded from this measurement. So after my more membrane-focused measurements, I turned my attention to the posterior chamber. So this section of the swim bladder was selected due to its importance in gas resorption, as all gases are released through the posterior chamber, through the oval, in a complex venous system. So the 3 posterior chamber measurements that I looked at were branching density, venous percent cover, and main vein diameter. These measurements were chosen because I believe they would have an effect on the efficiency of gas resorption and the speed at which gas could exit the swim bladder, especially during changes in pressure. During dissection, the posterior chamber was carefully separated from the rest of the swim bladder and placed in a glass dish until being photographed under a dissecting scope post-dissections. So I used Image J for all of my posterior chamber venous network measurements. So my first measurement was branching density. So I identified one of the main veins in the sample and measured the distance between 2 of the furthest discernible branches on the vein. So then I counted the number of branches between these 2 endpoints. And then to calculate the branching density, I divided the total number of branches by the vein distance. And then again, using Image J, a 14 by 14 grid was overlaid on each posterior chamber sample. With each row of 14 points, a random number generator was used to select 5 of these 14 points. So I looked at each of these points, and it was marked as having either any amount of contact with a venous structure or no contact. So the number of points that did overlay a vein were then added together. This value was then divided by 70 total points and multiplied by 100 to get the total percent cover. So next is main vein diameter. Within each posterior chamber sample, I identified the largest continuous vein. And using the vasometrics tool within Image J, I drew a line through the center of the main vein. The vasometric tool fits lines perpendicular to the center through line. So then all of these perpendicular lines all took diameter measurements of the vein, and these values were averaged together to get the diameter of the main vein. So then after taking all these measurements and compiling all my data, I used a combination of univariate and multivariate statistics to analyze my data. Univariate statistics included a chi-square, ANCOVA's logistic regressions, and multiple logistic regressions. These were used to identify specific relationships between one or two factors. I also ran a number of multivariate statistics, including NMDS and Permanovas, PCAs, GLMs, and binomial GLMs. These analyses were used to look at combinations of and interactions among multiple factors. So then moving on to my results. The next 4 slides pertain to Objective 1, or my habitat zone objective. Again, you can look up to the red rockfish to see where we are in the results. So a combination of 5 swim bladder morphological measurements were used to calculate a Bray-Curtis dissimilarity matrix, which was then used to create a non-metric multidimensional scaling, or NMDS plot. Within the plot, there was very little overlap between the 2 habitat zone ellipses, which means there isn't a lot of similarities between these swim bladder measurements. This separation is enforced by the Permanova results, which confirm that there are significant differences in swim bladder measurements between habitat zones. So generalized linear models, or GLMs, were used to investigate the effect of habitat zone on all 7 swim bladder measurements. Even though I used GLMs, these graphs show the results of t-tests, as they are easier to visualize while getting the same point across. Just as a reminder, the posterior chamber is important for its role in gas absorption, and these specific measurements may influence the rate and the efficiency of gas release. Despite identifying differences among habitat zones, the posterior chamber venous network measurements did not contribute to this variation, as there is no difference in any of the posterior measurements, and there is a lot of variability seen in each one. I then continued with habitat zone GLMs for additional swim bladder morphological measurements. Both tearing threshold and total membrane thickness were significantly different between habitat zones, with the swim bladders of benthic species requiring more pressure to tear and having thicker membranes than that of semipelagic species. And then I wanted to see which species were driving the differences seen in those 2 significant GLMs. So I ran a second round of GLMs, but replaced habitat zone with species in the model. I then followed the GLM with a Tukey's post hoc test to look at species pairwise comparisons. In both tearing threshold and total membrane thickness, the measurements for vermilion rockfish were significantly higher than most of the other species, which emphasizes the thick, robust morphology of vermilion swim bladders. So the next 2 slides focus on Objective 2, or the length objective, to investigate the effect of total length and species on each of the 7 swim bladder measurements, and ANCOVAs were run. So similar to the results seen with habitat zone, there is no effect of total length or species on any of the posterior chamber venous network measurements, with all 3 of these measurements showing a lot of variability with length. But total length did have a significant effect on tearing threshold, total membrane thickness, and posterior membrane thickness, with these measurements increasing with an increase in total length. Even though species didn't have a significant effect, many of the gopher rockfish measurements were higher than, or greater than, many of the blue rockfish measurements in all 3 of these morphological measurements. So the next 2 slides focus on Objective 3, which looked at the effect of swim bladder morphology on barotrauma. So I performed a principal component analysis, or a PCA, to determine the species level associations of a combination of 5 different swim bladder measurements. The PCA transformed the 3-dimensional swim bladder measurements to fewer dimensions, or axes. I then used these 2 axes as factors within a multiple linear regression. Anterior and posterior membrane thickness, venous diameter, and percent cover all loaded negatively on PC1, which explained 43.3% of the variance. And tensile stress loaded negatively on PC2, which explained 24.3% of the variance. And as you can see in the PCA biplot, there is a lot of separation among the species and between the ellipses. So to investigate the influence of swim bladder measurements on barotraumatic susceptibility, I used the principal component scores for PC axis 1, PC axis 2, and approximate capture depth as factors within a multiple linear regression with barotraumatic index as my response variable. So the overall model was significant, with barotraumatic index exhibiting a positive significant association with capture depth, meaning that fish caught deeper experienced more barotrauma injuries. But barotraumatic index showed no association with either PC1 or PC2, which had a lot of swim bladder morphology information on them. The next slides, multiple slides, obtain to objective 4, which is looking at the effect of other factors aside from swim bladder morphology on barotrauma. So during dissection, I identified if there was a tear in all of my fish. So using this information, I then ran a chi-square test and found that gopher rockfish experienced the highest incidence or the highest rate of swim bladder tears compared to the other 3 species, with approximately 54% of gopher rockfish identified as having a torn swim bladder. I then ran a logistic regression that looked at the relationship between capture depth and rupture probability in rockfish species, and I found that there is a significant relationship between capture depth and the probability of a torn swim bladder in blue rockfishes, meaning blues caught deeper were more likely to experience a swim bladder rupture during capture. And despite gopher rockfish experiencing a greater incidence of swim bladder tears than the other species, capture depth did not affect the occurrence of gophers experiencing tears, as they experienced tears at all depths, even as shallow as 18 meters. So I then used a binomial GLM to identify any significant associations between a variety of explanatory factors, including capture depth, total length, barotraumatic index, and habitat zone, and the probability of swim bladder tears. I found that benthic fishes and fish caught deeper were more likely to experience a swim bladder rupture than semipelagic fishes and fish caught shallower in the water column. I then ran an additional GLM to investigate the effect of barotrauma on species from different habitat zones. Benthic fishes experienced more and more severe barotraumatic injuries than semipelagic fishes, with both gopher and vermilion rockfishes having greater barotraumatic indices than blue and olive rockfishes. I then ran an ANCOVA to investigate the effect of total length and habitat zone on barotraumatic index. So in this model, there was no significant relationship between total length and barotraumatic index, but the incidence and severity of barotrauma decreased in larger semipelagic fishes. So finally, I ran a global model that incorporated 5 different life history characteristics to see which played the largest part in explaining barotrauma susceptibility. The explanatory factors included habitat zone, total length, sex, capture depth, and the interaction between capture depth and habitat. Ultimately, capture depth and the interaction between capture depth and habitat had a significant effect on barotraumatic index. Even though habitat zone was significant in my previous univariate ANCOVA, when incorporated with many other potential explanatory variables, it lost its significance. So by incorporating this multivariate analysis, it highlights that you can't really generalize across species, and the influence of different factors may change when looking at the bigger picture within the data. So now I'll wrap things up by digging deeper into my results and talking about the potential management implications of my findings. So starting with my habitat zone results, I hypothesized that benthic species would have greater tearing thresholds and thicker swim bladder membranes than semipelagic species, and my results found both of these predictions to be true. So benthic species spend the majority of their lives on the seafloor and rarely come up off the seafloor and don't really ever make any sort of vertical movement. So in general, benthic species evolved earlier than semipelagic species, and they have life history traits similar to that of some of the most ancestral rockfish species. And the thick, rigid swim bladders seen in this study seen in these benthic species are similar to what can be assumed to be the basal condition. So they've had a lot more time to become more narrowly adapted to their seafloor habitat. So continuing with habitat zone results, I hypothesized that semipelagic fishes would have more elastic swim bladders that could stretch further before tearing than benthic fishes. While this result wasn't significant, many semipelagic individuals had a higher tensile stress than benthic individuals. This result highlights the ability for semipelagic swim bladders to constantly adjust their size as they stretch and compress while undergoing changes of pressure during their vertical migrations. Other characteristics, such as a thinner swim bladder membrane, may have developed to assist with more efficient gas absorption rates, as semipelagic species need to frequently adjust their buoyancy within the water column. So multivariate statistics show differences in swim bladder morphology based on a variety of measurements. The separation of the ellipses in both of these graphs highlights these differences. If you look at the NMDS plot on the left, benthic species have thicker, what do they have? They have thicker anterior and posterior membranes and greater venous diameters, but lower tensile strength and less venous percent cover compared to semipelagic species. And if you look at the PCA plot on the right, the ellipses of blue and vermilion rockfishes have the most separation within the PC space, coinciding with the interspecific differences seen in membrane thickness and tensile stress. While these results are pretty definitive, an increase in sample sizes would hopefully solidify the distances between clusters of similar individuals. So in both of my first two objectives, there is no effect of habitat zone or total length on any of the posterior chamber venous network measurements. While I do think these swim bladder measurements may still play a role in gas absorption rates, I just wasn't able to get consistent enough measurements on a fine enough scale to identify differences among species. With these measurements, I was really limited by sample size, and despite having standard dissection protocols, there is so much inconsistency and variability among individuals. If I were to repeat this study, I would utilize techniques such as fixative perfusion and dye injection-based visualization in order to look at the fine capillary network at a microscale. So moving on to the total length results, I hypothesized that the swim bladders of larger rockfishes would have greater tearing thresholds and thicker membranes, both total and posterior chamber, than smaller rockfishes. And all three of these hypotheses were true and all had a significant positive relationship with total length. So larger fish have larger volume swim bladders in order to maintain buoyancy for a heavier individual. And with more time for a more complete morphological development, larger semipelagic individuals are less susceptible to barotrauma, as their swim bladders may be better suited for changes in pressure. But overall, capture depth had the greatest influence on both barotrauma susceptibility and probability of swim bladder rupture. So fishes that were caught deeper experienced a larger change in pressure when captured, and the gases in the swim bladder expanded and either displaced other internal organs within the internal cavity or caused a tear within the swim bladder membrane. This finding could have a direct effect on rockfish regulations. Shallower fishing may result in fewer barotraumatic injuries, fewer swim bladder tears, and an increased likelihood that discarded fish may be able to swim down to depth on their own. So looking at the bigger picture of the rockfish fishery, the population, mortality, and fishing yield undergo constant fluctuations based on a number of different factors. The maximum sustainable yield, or the MSY, is the highest possible annual catch that can be sustained over time, which includes total mortality within the fishery. So total mortality includes both natural mortality and fishing mortality, which is made up of discard mortality rates and retained fishes by anglers. So fishing mortality is a constant, so a reduction in discard mortality rates would theoretically result in increased catches for anglers. By decreasing discard mortality rates, we will be ensuring sustainability and longevity of the fishery. So in order to keep the rockfish fishery sustainable, the California Department of Fish and Wildlife could take two different approaches. The first approach would focus on reducing discard mortalities. In this study, which focused specifically on nearshore species, it was found that fishes that were caught deeper and smaller fishes experienced the highest degree of barotrauma. A potential solution would be to limit fishing to areas less than 50 meters, which would result in fewer fatal barotraumatic injuries. Additionally, you could potentially increase hook sizes, which would hopefully discourage smaller fishes who are more prone to barotrauma from biting the hook and being reeled up and potentially released. Lastly, while California just passed a law mandating the presence of descending devices on boats carrying any species of federally managed groundfish, this does not mean that they will be used. Descending devices have been proven effective at increasing survivorship of released fishes, so I believe that they should be used whenever a fish is not retained. If these practices aren't effective and we see declines in the population, further management actions may need to be taken. So a stricter approach would be to limit the overall number of fishes removed from the population. This can be done by decreasing the total number of rockfish that are able to be taken by each angler, or making sure the first X or any number of fish that are caught and retained, are retained, and that fishing stops as soon as an angler fills his or her bag limit. The second option would hopefully dissuade anglers from cherry picking their catch or trying to catch just one more big fish. There are other management options that limit the spatial and temporal aspects of the fisheries, but regulations like these are a hard sell for the fishing community, as it limits both the time you're able to fish and the number of fish you're able to keep, which some say are the best parts of fishing. And for anglers, it is hard to look at long-term goals when short-term solutions are this restrictive. So overall, this study was far from perfect. And even though I did get some really interesting and useful results, there are a few things that I would do differently if I were to do the study again. First of all, I would just include different species within the study. Even though the 4 species that I used are better than 2, blues and olives aren't representative of all semipelagic species, and vermilions and gophers aren't representative of all benthic species. From a phylogenetic standpoint, it would be interesting to include species with similar evolutions to the current study species, as well as some intermediates, in order to understand how evolutionary radiation affects swim bladder morphology. A second potential study from this project would be to relate gas resorption rates to posterior chamber venous network measurements. And potentially the use of a larger hyperbaric chamber with some sort of window would allow you to see how long it takes fish to reach neutral buoyancy after a change in pressure. The rate of gas release that you get from this could potentially be compared to fine-scale venous network measurements, and hopefully you'd be able to find some sort of correlation between the two. And lastly, with global warming and ocean acidification being the hot topic right now, it would be interesting to see how warming water temperatures and a decreased pH affect gas resorption rates within the swim bladder. Because the movement of gas molecules increased with increasing temperature, would gas secretion and resorption rates also change? Would rockfishes be able to off-gas faster and decrease barotrauma susceptibility? While I don't have the answer to any of these questions, I hope this study has given everyone insight into the nuances of rockfish morphology, and I'm excited to see where this research may go. So now I'd just like to thank a lot of people that have helped me throughout my time here. So first, I just want to thank my thesis committee. So Rick, you have been instrumental in shaping me and making me the scientist I am today. I definitely came in with no experience with rockfish at all, but through the different projects I've been able to work on, I feel like I've gained so much through my time here. And even though I'm a little bit stubborn when it comes to some things and set in my ways, you've really worked with me in a way that has really shaped me to become, I think, a really good researcher. And for that, I'm really appreciative. And Scott, you, like Rick, have so much fish knowledge, so it's been great to just chat with you and bounce ideas off of you through every aspect of my thesis. Also, thanks for always being my dive buddy in Catalina, even though I think I had the coolest project, so that's fine. But yeah, you've just been so great throughout my time here. always having your door open and always being willing to chat about anything. It's been really great, so thank you. And then to Gita. So Gita's the only non-fish person on this thesis, which was probably for the best. When you think about fish, it's very easy to get blinders on and not really think about the bigger picture. So Gita came in and made sure that everything was addressed properly, and she really helped me with my stats. I thought I was pretty good at stats before this, but like after going through this, I know there's so much more that I can learn. And Gita was so helpful walking me through some of my analyses and just helping me kind of fine-tune my thesis. So thank you, Gita. And I'd also like to thank all the boat captains, deckhands, and volunteer anglers that helped with my fish collections. Without them, I literally would have no fish for my thesis, as I cannot go out and catch all those fish myself. So they were awesome throughout this process. They were really interested in what I was doing, which really made me happy, and it showed that even in their livelihood, they still want to learn more about the rockfish they are catching. I also want to thank Joe Welsh of the Monterey Bay Aquarium. Like I said, he let me borrow these hyperbaric chambers for over a year. He came out with me 1 or 2 times to show me the rope, but then really trusted me to make sure everything ran properly for all time after that. And it was great to just go down and chat with him and kind of update him on where I am. So thanks, Joe, if you're listening. And then to Larry Allen, I met him at a conference this year, and he gave me all the rockfish icons used in this presentation. So I'll probably use them for every single presentation I do for the rest of my life. So thanks, Larry Allen. And then to everyone here at Moss Landing, the staff and community. It's a really special place here. There's so much cool science going on, and the relationships you're able to build with the people here has been really good. And then to the Fisheries and Conservation Biology Lab. This was a really fun lab to come into. I think everyone in the lab was so welcoming, and they just acted as such great role models in research and in science, and I could go to them with anything. They really helped me shape the beginning portions of my thesis, and they've just been great to work with whenever I have. So yeah. And then to everyone in the ichthyology lab, they've kind of adopted me this past year. It's been so great having a like-minded group of people all work together on different boat projects and on different dissection days. So that's been really fun. And I have had a few different funding sources throughout my time here. So the Kenneth H. Cole Graduate Scholar Award, the Dr. Earl H. Myers and Ethel M. Myers Oceanographic and Marine Biology Trust, 2 different MLML Wave Awards, and then the CSU Coast Travel Student Travel Award. This funding has definitely been instrumental in kind of helping me finish up this research here. So for that, I'm super grateful. And then there's a whole bunch of other people that I want to thank. So my family and my friends here and back on the East Coast. My mom and my sister flew all the way across the country just to hear me talk about some swim bladders. So that's pretty cool. Then everyone else listening back at home, thanks so much. Your support has been, it can't be reciprocated. It's been so helpful in kind of getting me through here. Yeah, so to the Moose and Mossies crew, you guys are the best. You've made my time out here so much fun, and so even if I leave and go back to the West Coast, I know we'll stay friends forever. That's non-negotiable. And then to Chris, who's also watching back on the East Coast, he probably knows as much for a non-science person, he probably knows as much as swim bladders as anyone in the room. He's heard me talk about it so much. So even from a distance, he's been through all the ups and downs with me. He's more than willing to listen to every rant I have and kind of celebrate every triumph with me. So again, his support has been so great, especially these past couple months, kind of getting me through this last final stretch. That has been awesome. And then to my cat Crouton. She's pretty cool. She's been a solid housemate this past 2 years. She has a lot of energy, even when I don't want her to. Yeah, but she's pretty good. But yeah, but just thank you to everyone. And now I can take some questions if anyone has any.
Questions?
Looks like a strobe light.
And we can get questions from the audience listening online as well.
Thanks, Molly. Thanks, Rick. It's really great to see this work that I've heard Rick talk about for a number of years with different students to making it to this point.
Yeah.
And I'm really interested in your suggestion or the limiting fishing to 50 meters or less, or increasing hook size. And I wondered if you could just talk a little bit about the depth, how you came to that depth. I mean, I understand the fish hook size, but what about the depth?
Yeah. So I found, not just in my project, but just in fishing in general, when you catch fish deeper than 50 meters, they come up with so many barotraumatic injuries. The rate of mortality of release fishes definitely increases once you pass that 50-meter mark. So there's just, I think in general, just a higher chance, a higher just survivorship at those shallower depths. Yeah. You don't have to.
Uh-oh, this will be the tough one.
So you want to limit the depth that fishermen are fishing
Yeah,
to 50 feet, but
50 meters.
if to 50 meters.
Yeah.
Are these fishermen trying to catch the biggest fish they can? I mean, is it their intent to catch them and then, eat them? And are they able to catch big fish at this shallower depth? So the depth doesn't determine the size of the fish. If you're going deeper, that doesn't mean necessarily you're going to catch a big fish.
Yeah, so I would say most fishermen probably want to catch the biggest fish they can, because a lot of people are just fishing to retain the fish and eat the fish and everything like that. Within 50 meters of fishing, you definitely can catch some larger fish. You also have smaller fish at those depths. I would say there probably are larger fish further offshore, as some of the bigger fish are those deep water slope and shelf species that you wouldn't catch near shore or closer to shore, potentially in this 50-meter range. But I still think you're able to catch pretty big fish within that that kind of restricted zone. Maybe not the biggest fish you'll ever catch in your life, but I think you'd be able to catch enough fish to, within a bag limit, to be pretty happy with what you get.
Thank you, Molly. I learned so much today.
Oh,
I'm curious about I didn't know how swim bladders worked, so thank
yeah.
you for introducing them.
Of
That was great.
course.
So I was thinking about diffusion and rates of diffusion, and is there work that's been done on the rate that they're brought up and whether that can have an influence? And likewise, do some of the bad effects mitigate over time if you keep a fish on deck?
Yeah, so that's a great question. When I talk to anglers about this, they're like, oh, but I feel like I reel in fish pretty slow. I feel like that should decrease how much barotrauma they're able to get. And there have been some studies, and it takes hours upon hours. In semipelagic species, there was a study, and it was to become neutrally buoyant after a change in 20 meters, it took 40-something hours. So people are like, oh, the 2 extra minutes that I reel in, it really doesn't make that much of a difference. Sorry, what was your second question?
Well, it's the flip side, so probably that answers the other question too, which is, can they equalize or equilibrate once they're on deck, if you keep them on deck for a while?
So, theoretically, yes, if you're able to keep them submerged in a controlled and contained environment. Most of the time, when fish come up, to increase survivorship, you want to get them off the deck and back down to depth in about 5 minutes. If you do happen to keep them, they're usually so positively buoyant that they'll be floating on top of whatever holding tank you have them in. And just the injuries they sustain really limit how long they're able to survive for. Yeah, for some of the semi-pelagic species, you're able to keep them as they're not as affected by barotrauma. Yeah, but some of those benthic species, it's hard to keep them alive without some sort of recompression or venting the swim bladder, which is not the best thing to do. Yeah.
But I'll add on to that by saying there is a paper that shows that the barotraumatic traumatic symptoms increase the longer you have it on board, so they're not decreasing after time.
Yeah, sometimes when we're working with fish and they'll be on a tag board and you like accidentally brush their eyes, it will light up with all the crystallization and everything like that. So they definitely can worsen with the time that they spend on deck. Yeah.
Hello.
Hello.
I was thinking as you were talking about like the torn swim bladders and the barotraumatic index, did you ever look to see if there was a relationship between whether the fish had a torn swim bladder and kind of the severity of barotrauma symptoms? I was just sort of thinking, like, as they're coming up, if the swim bladder ruptures, are they going to show less barotrauma or
no,
more?
There's no relationship. I looked at that. There's no significant relationship in benthic or semi-pelagic fishes. I thought there would be too, but yeah, that's what data says.
Great job, Molly.
Thank you.
So you came at this from, you talked a lot about like the phylogenetics of this and evolution.
Yep.
Have you thought about what impact the habitat that the fish grew up in, like after it settled out as a larvae, what impact that would have, especially on like the Venus network and like temperature and DO? What kind of impacts those might have?
That's a great question. I don't really know. So can you reword your question a little bit?
In what way?
So you're
just so like after the fish settle out
Yeah.
from their larval stage,
yeah.
if they're exposed to different temperatures or different dissolved oxygens, would they have more or less venation on the swim bladder? Would the swim bladder have different morphologies as opposed to it being strictly evolutionary?
I think that's the next tent project. I think they really want to look at that and look at the fine scale Venus measurements of larval fishes. That sounds wonderful. I don't really know. I'm assuming it will have more of a phylogenetic impact than like exposed to different treatments. I don't think they would have like greater percent cover or branching density or anything like that when exposed to different treatments. I think it's more of genetic and phylogenetic based, but that would be really interesting. I think it would be really tough to quantify on that, or these measurements on that small of a scale. But that would be interesting to do if someone wanted to do it and and they have really good microscope skills. I don't think I have those. So, yeah, that's a good question though.
It was a really great presentation. Thank you.
Thank you.
So with the swim bladder, is the elasticity of the swim bladder, like the manner in which it inflates, is it directional? Does like when you think about a swim bladder barotrauma, you think about it like coming out of its mouth, which would be kind of like a forward-backward inflation direction, I suppose, because swim bladders.
So it's the esophagus or the stomach coming out of the mouth.
Oh, okay, got it. So it's pushed out.
Yep.
Does the swim bladder like inflate equally in all directions?
I think it does, yeah. It is fused kind of to the rib cage within the internal cavity, so kind of probably moving. It's probably limited in its movement, like up towards the dorsal surface, so it obviously moves kind of out towards the stomach a little more, but I haven't really seen if it's moved or like expanded more latitudinally or longitudinally along those two aspects. I would assume it's kind of the same in all directions.
Thank you.
First of all, great job.
Thank you.
I was wondering, you mentioned maturity early on, and I was curious if you had any thoughts about how maturity and stage, like of the fish, would affect swim bladder morphology. Like if it's sexually mature, is it going to put more energy into reproduction versus, you know, making the swim bladder thicker and, you know, over time rather than just age, but like specifically sexual maturity?
Yeah, that's a good question. I didn't really see any differences in measurements among males and female or between males and females. I think a few other papers also found that same result. It'd be interesting, yeah, if you're somehow able to look at energy expenditure and what they kind of allocate their energy towards. That would be really interesting. And I think like from a morphological standpoint as well, obviously with these females you have these large ovaries, and kind of seeing how that also affects where the swim bladder is able to move in the internal cavity. Yeah, I definitely think, actually I don't definitely think, maybe size has more of an effect than actual maturity, but that would be something really interesting to look at, yeah.
Okay. Does anyone know if we have any questions from the online? We don't. So we're going to take her away and put the screws on her for a few minutes, and then we'll come back and celebrate. So thank you all. One more round of applause for